is titration? Titration is a quantitative chemical analysis technique that is used to determine the concentration of a substance in a solution. It is based on a complete chemical reaction between the substance, known as analyte, and a reagent known as titrant. The endpoint of the titration is typically detected using an indicator, which changes color when the titration reaction is complete. The main purpose of titration is to determine the unknown concentration of an identified analyte in a sample. Titration allows for precise measurement of the concentration of a substances in a sample. Other than that, the purpose of titration is to ensure quality in industry by checking the composition of product. Titration is known for its accuracy when performed correctly. It enables researchers and analysts to determine concentration with high precision. Overall, the purpose of titration is to provide accurate and reliable information about the concentration of substances in solution, which is crucial for understanding chemical processes, maintaining product quality, and advancing scientific knowledge.
Next, I will talk about example of titration. First is acid-base titration. Acid-base titration is involved the reaction between an acid and a base. The equivalent point is often indicated by a pH indicator or a pH meter. This type of titration is applied when measuring the acidity of a product. The example is, the acidity of vinegar is determined by titration with a base, example, sodium hydroxide. This helps ensure that vinegar meets the required standards and tastes consistent. Second one is precipitation titration. It involves the formation of a precipitate during the reaction. Indicators that change color from precipitation can be used. This type of titration is applied in quantifying halide ions in pharmaceutical compounds. Precipitation titration is used to quantify halide ions, chloride, bromide, and iodide in various pharmaceutical preparation. This is crucial for ensuring the correct formulation and stability of the drugs. Last one, redox titration. Redox titration is a type of titration based on a redox reaction between the titrant and the analyte. This titration is applied when determining the concentration of sodium hypochlorite in commercial bleach. The example is, the active ingredient in bleach, sodium hypochlorite, is quantified using redox titration. Potassium iodide is added to the bleach solution, liberating iodine. The iodine is then titrated with sodium thiosulfate, with the endpoint indicated by the disappearance of the yellow iodine color or using a starch indicator.
For this experiment, the required material is sample, which is orange juice, distilled water, iodine solution, beaker, aluminium flask, and also starch solution. The first procedure is put 5 ml of orange juice into aluminium flask. Second, we need to dilute orange juice, which is the sample, with 150 pure water. Third, we need to add 4 until 5 ml starch to the diluted orange juice, which is sample. Next, we need to fill 50 ml of urate with iodine solution. Last but not least, titrate the sample with iodine solution. When the titration comes to the end point, the solution of the color changes from clear into blue.
Next, I will explain about the results for determination of vitamin C in orange juice, this is one of the application of titration that we chosen in this topic. So the data in this application consists of standardizing the iodine solution and the ascorbic acid content in orange juice. So first is the data table 1, which consists of standardizing the iodine solution. Through this titration, the amount of ascorbic acid contained in 1 ml of iodine was 0.91 mg. This 0.19 mg of ascorbic acid is used to determine the value of ascorbic acid contained in the orange juice. Next, table 2. The data shows the value of titration in order to determine the value of ascorbic acid content in orange juice. In this titration, we use 10 ml of orange juice for each titration. After the result obtained, average amount of ascorbic acid that contained in the 100 ml orange juice was 25.175 mg over ml.
Here are some precautionary steps that we need to take while doing titration. First, hand must be washed after handling strong chemicals like strong acids to prevent erosion. Second, the sample must be stored in a dry place to avoid moisture affecting endpoint. Third, the sample must be kept at room temperature to prevent from the sample took long time to reach a set value during the titration. Fourth, all the readings on the gadget must be ensured that has been calibrated with inside the test to identify whether the equipment can be operated or not. And lastly, all the waste materials inclusive of used answer and the leftover samples must be disposed in correct disposal ways. Titration is an analytical technique that has been used in various industries due to its ability to determine the concentration of a substance in a solution accurately. For example, titration has been used in food and beverage industry to determine the pH of acidity of the beverages, the concentration of preservative, and vitamin concentration on food products. Other than that, cosmetic industry also use titration to determine the concentration of active ingredients and ensure the effectiveness of the product. Lastly, environmental analysis has largely using titration techniques to determine whether is there any pollutants in the environmental samples or not.
In conclusion, overall, what was previously discussed can teach us about the titration process and its significance. Members of this group had talked about the components involved in the titration process. Additionally, we present examples of realistic titration applications of titration on the methods and how to achieve the best results. That's all from us. Thank you.